Hi, everyone. So my name is Andriele Silva. I am a biochemistry PhD candidate at Brooklyn College in New York. And today I'll present my research entitled an in-silico approach to analyze and characterize the interaction of CD6 with CLAB2. So just a little background on my study. My project involves the cell cycle in budding yeast. And as you know, cell cycle is accomplished through a series of events that take place in a cell and cause it to divide into two daughter cells. So in budding yeast, Saccharomyces cerevisiae, it starts at phase 2. Here we have cell growth and some proteins get produced. Then in S phase, we have DNA replication. We move on to get phase 2, and we got more growth and more proteins are synthesized. And then in mitosis, sure. And then in mitosis, there is nuclear division, and that is accomplished in 4 phases: prophase, metaphase, anaphase, and telophase. Finally, we have the physical separation of the cells and cytokinesis. Our protein of interest is CDC6, so cell division cycle 6. It is necessary to promote pre-replication complex assembly in G1 phase, and that leads to helicase loading. So the first step of the pre-replication complex is recognition of the origin DNAs by ORC. So ORC is origin recognition complex. Now ORC recruits our protein of interest, CDC6, and then together they can recruit another complex, which is between the MCM27 helicases and CDT1, which is a licensing factor. We need ATP hydrolysis so that we can get a double hexamer of these MCM27 proteins, and they interact via their N terminals in a head-to-head fashion so that they can go in bidirectional way. So here in G1, we only have helicase loading. We don't have helicase activation until we reach S phase. So, 1 second. Okay. Now moving to mitosis, we have phosphorylation of CDC6 by a protein called CDK1. It phosphorylates CDC6 and terminus. There you see T7 and T23. That leads CDC6 to bind to CLAB2. Now, CLAB2 is the main mitotic cycling, and this binding of CDC6 to CLAB2 happens via this motif, LXF, and that inhibits CDC6 from origin licensings, that prevents DNA rereplication, and that leads to, ensuring proper cell division. So this is a very important regulation control here. Now, the need for these studies. We wanted to do docking, we wanted to do some molecular dynamic studies, but we noticed that in the PD database, we did not have a high-quality CDC6 structure. We also did not have high-quality CLAB2 structures in the PDB database, and also not much is known about the molecular mechanisms of CDC6 and CLAB2. This is the experimental methodology we put together so that we could accomplish that. We first began by looking for templates, and we, in the case we found good templates, we followed homology modeling. So we used Mulder, for example, to model our proteins. If we didn't find any suitable templates, then we went the ab initio route. We used programs such as Quark, for example. And once we got our models, then we proceeded to model evaluation using 4 different modeling tools. Sometimes the models needed refinement, sometimes they didn't. And then for the docking, we used usually ClusPro and HDoc, and we analyzed the docking results with PyMol. And then we did mutation studies on key residues that were based on these docking results, and for that we used PyMol and Chimera. We then proceeded to compare the wild-type CDC6 CLAB2 complex to the mutated CDC6 CLAB2 complex using with GROMACS by doing steered molecular dynamic simulations. So here we move to our results. This is our full-length CD6 protein structure. It was very challenging to model this protein because it has several and very long intrinsically disordered regions in its N-terminal. And we indeed compared our model to AlphaFold because it was released just after we finished model this, and we saw that it scored much better in this. Our model scored much better, but I will show you how we found that out. So in this orange region here, this is how we modeled it separately from the rest of the protein. We use ab initio tools to model that. And in this region, we see that functionally important residues or motifs, such as the LXF motif, the N-terminal phosphodigrons, and the RXL motifs are all in this region. And then the C-terminal phosphodigrons for CDC6 are all in the well-structured region of this protein. So as I said before, we noticed that our model scored better than AlphaFold, especially in these intrinsically disordered regions. So AlphaFold has its own way of evaluating proteins, and you can see the orange intrinsically disordered prediction of AlphaFold. Now, we use several evaluation tools to compare our models, and we looked specifically for the intrinsically disordered regions because we wanted to see how our model did in comparison to AlphaFold. And just one example is verified 3D here. You have a threshold for model quality at 0.2, and both our N-terminal phosphodigrons and C-terminal phosphodigrons scored at 0.2 or higher. So that shows that our model scored better in that region. Then we model CLEB2, which also has several IDRs. It's very common within cell cycle proteins. And here it shows that both CAM box over here and D box, they're present in this IDR region of CLEB2. Now, I think both here in the CDC6, you can see that part of this IDR takes this alpha helix shape, and we believe that's a transient nature of the IDR that could take place upon binding. To model this protein, we took a similar approach to model CDC6. We modeled the intrinsically disordered region separately, and then we just incorporated into a previous model that we had. The hydrophobic patch is very important in this protein. It's a hub for protein-to-protein interactions, and you can see here that it falls in a very well-structured region of this protein. So using the two models that we generated, we were able to predict a potential scenario of CDC6 and CLEB2 interaction, where CDC6 glutamate 45 interacts with CLEB2 lysine 270, which is located in the hydrophobic patch. We speculate that a transiently structured helix adopted by the PEC LXF motif in our CD6 model may be important for this interaction. And also, this is very interesting because we collaborate with a lab, Dr. IQA lab, and in their model, in their experiments, they see that the phosphodigrons in this interaction, they are protected by CLEB2, and we also are able to see this in our model, which is pretty neat. So when we were doing this study, we started to think of the importance of this glutamate 45 because although it's in the LXF motif, it's just one residue before the LXF motif. So we wanted to see if this is actually relevant, if it's something that is repetitive in cell cycle protein interactions with other cell cycle proteins. So then we were looking for a protein that had the same characteristics, for example, as CDC6. So we found BUD3, which is important in budding yeast because it localizes with septins to the bud neck. Contractile ring mitosis is also involved in actual cellular bud site selection. And BUD3 is very important because it's a known substrate of CLEB2. And it also has that glutamate right before the LXF motif. So this protein is a very large protein, over 1,600 amino acids, and it took a long time for us to model this protein. And we used a similar approach. We used a lot of modeler advanced scripts to model this one. And then after we got a very good model, we did the docking, and we saw that a very similar pattern happens here. So again, the glutamate, which is the equivalent, the glutamate here is 1631, but it's equivalent to the glutamate at 66, interacts with lysine 270 in its hydrophobic patch. So we see a similar binding pattern here, and we want to investigate other proteins to see if this continues to repeat. So this is something I'm very new to, and I actually start learning in June in a workshop. And we decided to try to look at the stability and do some mutation studies here on that glutamate 45 to see if something happens to our protein, if folding gets disrupted, and also look into binding energies, which we are working right now. So the first steered molecular dynamics simulations we did using GROMACS was for the wild type CDC6 CLEP2 complex. So in here, hopefully it plays. Okay. Is it working? Okay, so in here you can see that the proteins are being pulled apart. We want to see the force on how they get pulled apart so that we can understand the binding affinity here. And then we got a pull force plot that shows us how much strength you need to pull them apart, right? So the calculation estimated that it takes around 900 kilojoules per mole to pull the wild type CDC6 from CLEP2. Then we did the same thing, but to a mutated CDC6, where we changed glutamate 45 to alanine. And then we did the simulations under the same conditions. And when we did the simulations, you already see that the protein CDC6, which is to my left, you can see that it's already not as folded. The protein binding is not as tight as it was previously. And also when you look at the pull force, it is less pull force, the calculation is less. So it kind of shows that the stability is a little bit disrupted, and also it doesn't take as much energy to disrupt this binding. So we believe that wild type CDC6 with CLEP2, it has a better affinity. So our conclusions is that CDC6 and CLEP2 contain intrinsically disordered regions that may assume enough a helical structure upon interaction with other proteins. We got the CDC6 glutamate 45 interacting with lysine 270 from CLEP2. Then we did the BUT3 CLEP2 interaction, showing a similar mechanism to CDC6 CLEP2 interaction. And then we saw the stereoMG comparison, which showing that the wild type CDC6 CLEP2 complex has, better binding affinity than the mutated one. So I'd like to thank you all for watching my presentation. The organizers, my PI is here, which is incredible. And yeah, thank you. I'll take any questions.
Thank you, Andriele. Yeah, we have time for a couple of questions. I'll start with mine. So I wonder how conserved is that hydrophobic patch you're finding? Like, is it the same sequence in all of the Saccharomyces, or you find, because it's well known that the cell cycle in Saccharomyces cerevisiae is found to be variable? Is it related to this?
Yeah, that's a very good one. Yeah, it's very conserved. And there are many studies showing that specifically the lysine 270 is a very key residue in protein-to-protein interaction, even in other organisms.
So there are not variable sites within Saccharomyces in this area?
Mm-hmm. Absolutely.
Is there any other question?